All right, so today we're taking a deep dive into the fascinating world of molecularly imprinted polymers, or MIPs.
Catchy name, right?
It is, and I'm already intrigued just from the notes you sent. These MIPs sound like something straight out of science fiction, like tailor-made sensors at a molecular level.
That's a pretty good way to put it. The concept is surprisingly simple, yet incredibly powerful.
Okay, well, break it down for me then. How are these MIPs actually made?
Imagine you have a mold, like the kind you'd use to make a clay sculpture.
Okay, got it.
But in the case of MIPs, our mold is actually a molecule, the very molecule we want to detect.
So you're using the target molecule itself as a template to create a sensor that's perfectly designed to bind to it.
Exactly. We start with a mixture of monomers, which are like the building blocks of polymers, and we introduce our target molecule into this mixture.
So it's like mixing the ingredients for a cake, but instead of flour and sugar, you're using these monomers, and your special ingredient is the target molecule.
I like the analogy. Then we initiate a polymerization reaction, which causes the monomers to link together and form a polymer network. And here's the key. As the polymer forms, it grows around the target molecule, taking its shape.
So the target molecule is essentially guiding the construction of its own binding site within the polymer.
Exactly. Once the polymerization is complete, we're left with a solid polymer matrix that has the target molecule embedded within it.
Okay, so you've got this solid block of polymer with the target molecule trapped inside. Now what?
Now comes the imprinting part. We remove the target molecule from the polymer matrix, leaving behind a cavity that's perfectly shaped to rebind to that specific molecule.
It's like taking the cake out of the pan, right? You're left with a mold, but in this case, it's a molecular mold that can recognize and bind to our target.
Precisely. And that's what makes MIPs so powerful. They offer a level of selectivity that's incredibly valuable in a wide range of applications, from environmental monitoring to medical diagnostics.
Now, you mentioned selectivity, and I know from your notes that this is a big advantage of MIPs over traditional biological sensors like antibodies. Can you expand on that a bit?
Think about the environments where you might want to use a sensor. They're often exposed to harsh conditions, extreme temperatures, varying pH levels, you name it. Antibodies are sensitive to these changes. They can degrade and lose their effectiveness.
So they're a bit fragile.
In a way, yes, but MIPs, on the other hand, are synthesized from much more durable materials. They can handle a wider range of conditions without breaking down or losing their selectivity.
So it's not just about grabbing onto the right molecule, but doing it reliably, even in tough environments.
Precisely. And if we consider the cost factor, well, MIPs are significantly cheaper to produce than antibodies, which opens up many possibilities for using them in large-scale applications where cost-effectiveness is crucial.
Makes sense. So cheaper, tougher, and just as good at grabbing onto those target molecules. That's a winning combination.
Absolutely. And this is where we start to see the real-world impact of MIPs. One area where they're making waves is in environmental monitoring.
Right. Detecting pollutants in water, for example. It's almost like having these tiny molecular bloodhounds sniffing out specific contaminants.
Great analogy. Imagine deploying MIPs to monitor water quality in rivers and lakes. They could be constantly on the lookout for specific pesticides, heavy metals, or anything else that could harm the ecosystem.
So instead of taking water samples and sending them to a lab, we could have these MIPs out in the field constantly providing real-time data on pollution levels.
Exactly. And that real-time monitoring could be a game changer for environmental protection. It would allow for quick responses to contamination events, potentially preventing major ecological damage.
It's not just reacting to pollution, it's about getting ahead of it. You could use MIPs to proactively monitor areas that are at risk of contamination, right?
Absolutely. We could strategically place them near industrial sites or agricultural areas to ensure we're staying ahead of any potential problems.
This is all incredibly impressive, but it's clear that environmental monitoring is just one piece of the puzzle. You mentioned medical diagnostics, and I'm curious to dive into that a little deeper.
Sure. Think about the potential of using MIPs to detect specific biomarkers for diseases.
Okay, like a blood test, but much faster and more targeted.
Exactly. You could create a simple test that could quickly identify the presence of a particular disease, maybe even something you could use at home.
So faster diagnoses, potentially even before you show symptoms. That could be life-saving for certain conditions.
Absolutely. And for many diseases, early detection is key to successful treatment. So not just detecting illness, but paving the way for more effective interventions.
It sounds like MIPs are not just about fancy chemistry. They have the potential to make a real difference in people's lives.
That's what makes this field so fascinating. It's where cutting-edge science meets real-world applications with the potential to make a significant impact.
Now, I'm curious about how specific these MIPs can be. We've talked about how they can target certain molecules, but can they really distinguish between molecules that are very similar in structure?
Oh, absolutely. Their selectivity can be incredibly precise, able to differentiate even between molecules that differ by just a single atom or a slight change in shape.
Wow, that's pretty impressive. So it's not just recognizing a general class of molecules, it's about picking out a very specific target from a complex mixture.
Exactly. And this level of precision opens up incredible possibilities for things like drug targeting and environmental remediation. Imagine being able to design MIPs that can selectively bind to and remove specific pollutants, even in the presence of other similar molecules.
It's like having a molecular scalpel. Amazing. But I have to ask, are there any downsides to using MIPs? I imagine there must be some challenges or limitations.
You're right, there are. One challenge is achieving that high level of selectivity and binding affinity consistently for every target molecule. Sometimes the complexity of the target molecule structure makes it difficult to create MIPs that work as well as we'd like.
I imagine larger, more complex molecules would be harder to create MIPs for, right?
That's definitely a factor. Designing MIPs for bigger, more intricate molecules can be more challenging because there are more interactions to consider during the imprinting process. It's like trying to build a complex Lego structure with your eyes closed. It takes a lot of finesse and fine-tuning.
So it's not a guaranteed success every time. There's still a lot of research and development needed to optimize these MIPs.
Absolutely. And that's what makes this field so dynamic and exciting. Scientists are constantly exploring new techniques and approaches to improve the performance of MIPs. It's a field that's rapidly evolving with each new discovery.
Well, it's been an eye-opening journey so far, learning about how these MIPs are made, the challenges researchers face, and the potential impact they can have across so many different fields. I'm already eager to dive deeper into specific applications in our next segment.
Me too. There's so much more to uncover about these remarkable molecular detectives.
All right, welcome back. I'm still amazed by how versatile these MIPs are, from environmental monitoring to medical diagnostics. It seems like there's no limit to their potential applications.
And to think it all started with a simple yet brilliant idea taking inspiration from nature.
Oh, you mean like biomimicry, where we try to imitate nature's designs?
Exactly. Nature has had billions of years to perfect its designs. We'd be foolish not to try to learn from those designs.
So where does nature fit into the story of MIPs?
Well, MIPs are essentially synthetic mimics of antibodies, those remarkable proteins our immune system uses to fight off infections.
Ah, that makes sense. Antibodies are incredibly specific, aren't they? They can recognize and wind to particular foreign invaders, ignoring everything else.
Precisely. And that's exactly what we're trying to achieve with MIPs, creating synthetic materials with that same level of specificity.
So by studying antibodies, scientists got the idea for creating MIPs. That's pretty cool. It shows how understanding one area of science can lead to breakthroughs in a completely different field.
Absolutely. It's a testament to the interconnectedness of knowledge and the power of interdisciplinary research.
Now, I'm curious about the different types of MIPs being developed. Are there different approaches to creating these molecular molds?
Yes, definitely. There are several approaches, each with its own advantages and disadvantages.
This is what I love about these deep dives. Always something new to learn. So walk me through some of these different approaches.
One common method is called covalent imprinting. In this approach, the template molecule is actually chemically bonded to the monomers during the polymerization process.
So the template becomes part of the polymer structure itself.
Exactly. Once the polymer is formed, the template is then cleaved off, leaving behind a cavity with the precise shape and chemical functionality to rebind to that template molecule.
Sounds pretty straightforward. But are there any downsides to this method?
Well, one potential drawback is that those covalent bonds can be tough to break. It can make removing the template quite challenging and sometimes affect the MIP's binding capacity.
I see. So you might end up with fewer available binding sites, which could make the sensor less sensitive.
Right, there's always a bit of a trade-off. You want a precisely imprinted cavity, but you also need to be able to remove the template easily.
So are there any other approaches that might address this challenge of template removing?
Another common method is non-covalent imprinting. In this case, the template molecule isn't directly bonded to the monomers. Instead, they interact through weaker forces like hydrogen bonding or van der Waals forces.
Okay, so it's more like the template is guiding the polymer assembly rather than being directly attached to it.
Exactly. And because those interactions are weaker, removing the template is typically much easier. However, the downside is that the resulting cavities might not be as well defined, which can potentially impact the selectivity of the MIP.
So again, it's a trade-off, precision versus ease of template removal.
Right. And scientists are always exploring new variations and techniques to fine-tune these approaches and create MIPs that are perfectly suited for specific applications.
It's fascinating to see how this field is constantly pushing the boundaries, always innovating and refining these techniques. So we've talked about how MIPs are made, but I'm eager to learn more about where they're actually being used. What are some specific areas where MIPs are making a real impact?
Well, one area that's already seeing significant benefits is food safety testing. MIPs can detect contaminants like pesticides or toxins in our food, ensuring what we eat is safe.
So those molecular detectives are working hard to keep our food supply safe. That's reassuring.
They are, and they do it with incredible sensitivity and selectivity. It's a real game changer for food safety testing.
Food safety is something we often take for granted, but it's crucial for public health. It's great to see these MIPs playing a role in keeping us all safe.
Absolutely. And speaking of health, another exciting area for MIP applications is in pharmaceutical analysis.
That sounds intriguing. How are they used in that context?
MIPs can monitor drug concentrations in biological fluids, ensuring patients receive the right dosage and minimizing the risk of harmful side effects.
So they're like personalized drug monitoring systems, ensuring that medications are working safely and effectively.
Exactly. This is particularly important for drugs with a narrow therapeutic window, where even small variations in dosage can have significant consequences. MIPs allow for precise measurements, helping doctors to fine-tune treatment plans and optimize outcomes for their patients.
It's remarkable how these molecular sensors can provide such detailed information about what's happening inside the body. It's like opening a window into the intricate world of our cells.
And that's just the beginning. Researchers are even exploring the use of MIPs for targeted drug delivery, where they could be used to carry medications directly to specific cells or tissues, reducing side effects and improving treatment efficacy.
So instead of flooding the entire body with a drug, you could use MIPs to deliver it precisely where it's needed.
Exactly. Think about the possibilities for treating diseases like cancer, where you could deliver chemotherapy directly to the tumor cells, sparing healthy tissue.
It's incredible to think about the potential of MIPs to personalize medicine and revolutionize how we treat diseases. But I want to circle back to environmental monitoring for a moment. We've talked about how MIPs could be used to detect pollutants in water, but what about air quality? Can they be used to monitor that as well?
Absolutely. MIPs can be designed to target a wide range of pollutants, including those found in the air, such as volatile organic compounds or even specific gases that contribute to air pollution.
So they could be used to monitor air quality in urban areas or industrial sites, ensuring a healthy environment for everyone.
Exactly. And remember that incredible selectivity we talked about? That allows MIPs to pinpoint the presence of specific contaminants, even in very low concentrations, providing valuable data for air quality management and pollution control efforts.
It's like having a network of tiny air quality inspectors constantly monitoring the environment. It's amazing how MIPs can provide such precise and detailed information about our surroundings.
And that data is essential for making informed decisions about environmental regulations, cleanup efforts, and sustainable practices.
It's truly inspiring to see how these tiny molecules are having such a big impact on some of the most critical challenges facing humanity. From ensuring food safety to protecting our environment, MIPs are making a real difference.
And as MIP technology continues to evolve, we can expect to see even more innovative applications emerge. This is a field with immense potential, and I believe we're just scratching the surface of what's possible.
All right, welcome back. I feel like we've just scratched the surface of the incredible world of MIPs. I mean, where do we even go from here? Self-healing materials, personalized medicine, it's mind-boggling.
It is exciting, isn't it? And honestly, what we've discussed so far is just the tip of the iceberg. The potential of MIPs is truly vast, and I have no doubt we'll continue to see amazing breakthroughs in the coming years.
Well, you've got me hooked. So looking ahead, what are some of the most exciting frontiers in MIP research right now? What are scientists really focused on?
One area that has immense potential is personalized medicine. Imagine a future where we have wearable sensors that can monitor your health in real time, detecting even the slightest changes in your body's chemistry.
Wow, like having a personal health detective constantly on the lookout for any signs of trouble? That's incredible.
Exactly. These sensors could track biomarkers for all sorts of conditions, from heart disease and diabetes to cancer and infections. Early detection is so crucial for many illnesses, and MIP-based sensors could be a game changer, empowering people to take control of their own health.
It's a shift from reactive medicine to proactive healthcare, where you're staying ahead of potential problems instead of just treating them after they arise.
Right, and not just monitoring, but prediction as well. MIPs could help determine an individual's susceptibility to certain diseases based on their genetics and lifestyle.
So truly personalized healthcare, tailored to each person's unique needs and risks.
Precisely. And MIPs, with their incredible selectivity, are perfect for this. They can differentiate between subtle variations in molecular structure, providing a level of detail that's simply unmatched by other sensing technologies.
It's remarkable how these tiny molecules can provide such a deep understanding of our own bodies. But let's not forget about the potential of MIPs for drug delivery, which we touched on earlier. I mean, delivering medication directly to the source of the problem, that's got to be a game changer, right?
Absolutely. Imagine treating cancer by delivering chemotherapy drugs directly to the tumor cells while minimizing damage to healthy tissue.
That would revolutionize cancer treatment, reducing side effects, and improving patient outcomes.
Exactly. And that's just one example. The possibilities for targeted drug delivery with MIPs are immense.
It's truly amazing to think about how these tiny molecules are poised to transform so many aspects of our lives. From personalized medicine to environmental remediation, the potential impact of MIPs is truly global in scale.
And it's all thanks to the tireless efforts of researchers who are pushing the boundaries of this field, constantly innovating and discovering new applications.
It's truly inspiring to see science in action, solving real-world problems and improving lives. But of course, as with any powerful technology, we need to be mindful of potential risks and ethical considerations.
That's a crucial point. As we embrace the incredible possibilities of MIPs, we also need to proceed with caution and ensure that they are used responsibly and ethically.
For example, we talked about the pientry use of MIPs in surveillance technologies, which raises concerns about privacy and civil liberties. How can we ensure that this technology isn't misused in ways that infringe on individual rights?
Well, open and honest dialogue is crucial. We need to have transparent discussions about the potential implications of this technology involving policymakers, ethicists, and the public. Clear guidelines and regulations are essential, as well as robust oversight to prevent any misuse.
It's about finding the right balance between innovation and responsibility, ensuring that technological advancements align with societal values and ethical principles.
Exactly. And we also need to be mindful of the environmental impact of MIPs. As we scale up production and use, we must prioritize sustainability and ensure that these materials don't pose a threat to ecosystems or wildlife.
So it's about responsible innovation, developing eco-friendly MIPs, using sustainable manufacturing practices, and ensuring their safe disposal.
Absolutely. It's about embracing a holistic approach where we consider the long-term implications of our actions and strive to create a better future for generations to come.
Well, we've covered a lot of ground in this deep dive, from the fascinating science behind MIPs to their mind-blowing potential applications and the importance of addressing ethical and environmental considerations. It's been an incredible journey into the world of molecular design and a glimpse into a future shaped by these remarkable molecules.
It has been a pleasure discussing this with you. I hope our listeners are leaving with a sense of wonder and excitement about the possibilities that lie ahead.
And a sense of responsibility as well. The future of MIP technology is in our hands, and it's up to all of us to shape that future in a way that benefits humanity and protects our planet.
It's a future where even the tiniest of molecules can have a profound impact.
And on that note, we'll bring this deep dive into the world of molecularly imprinted polymers to a close. Thanks for joining us on this incredible journey of discovery.
Until next time, keep exploring, keep questioning, and never stop wondering about the amazing things that science can achieve.